Dear colleague, as shown by the list of courses you can read on this first slide, the fish-eDNA metabarcoding survey of the Danube, a part of the general eDNA survey presented by Alexander just before. This fish-eDNA survey has been carried out in collaboration with colleagues from several Danube countries who participate to sampling or any other operation. In addition to the collaboration between the ICPDR and DNAquanet, the Interact project measures allowed us to increase the number of sampling sites from 20 to 47 sites. 29 sites, the red circles, were sampled regularly along the Danube, and 18 on the main tributaries, the black triangle and the red circle for the Danube. For the tributaries, the samples were taken 5 to 10 kilometers upstream from their confluence with the Danube itself. All these sites were visited between June 29th and July 19th, 2019, during the GS4 survey. Water samples were taken at each site from a boat going along the entire cross-section of the river. The water is collected via a peristaltic pump, which is equipped with a disposable sterile tubing change for each water sample. The water is directly filtrated on the boat thanks to a cross-filtration capsule of 0.45 microns porosity. After filtration, the capsule is filled with a conservation buffer and can be kept at room temperature before DNA extraction. 2 water samples are collected at each site for a total volume of water varying between 8 and 82 liters. At the laboratory and after extraction of the eDNA, the eDNA is amplified using the universal primer Teleo and 50 cycles per PCR. 12 PCR are performed per sample, which means 24 PCR per site. After the final taxonomical assignment, 86 taxa were detected. 60 of them are well known from the Danube catchment, and 50 taxa from these 60 are identified at the species level, and 10 others at the genus 5 taxa or higher taxonomical level 5 taxa. All these 10 taxa group correspond to 24 species known from the Danube catchment. 5 taxa were unknown from the catchment. These false misallocations are mainly due to insufficient knowledge of haplotypes variability in the catchment, and an improvement of the reference database is needed for this taxa. 21 taxa were food fish, aquarium fish, and local farm fish, which could be detected in wastewater release to the river. Among these 21 taxa, 16 species are marine species and are well known as food fish like migre, herring, sea bass, mussel, tuna, sea bream, etc. They were mainly detected downstream at the vicinity of very large cities like Vienna, for example, you have on the Danube itself in red, or downstream from Bucharest on the Argeș River, 50 kilometers downstream Bucharest in blue, or also the Rusin-Kilom River. The Rusin-Kilom River is also well known as a very polluted river, and it appears that with this example it shows that fish-eDNA can be used to trace the sewage within rivers, even when the dilution factor is very high, like the Danube in Vienna. A traditional fish survey using electro-fishing method has been realized in parallel to the fish-eDNA survey, and the two methods can be compared at 26 common sites. 60 taxa have been detected by eDNA and 68 species caught by electro-fishing. 51 species are common to the two methods. 5 species have been only caught by electro-fishing, mainly due to the lack of species reference sequences in our database. 9 species were only caught by eDNA metabarcoding, and a large number are endangered or species of conservation interest. When comparing the species richness per site, the richness is significantly higher with eDNA compared to electro-fishing, even if some species from the same genera are not differentiated with eDNA. The mean richness is 29 taxa for eDNA per site, against 23 taxa for traditional electro-fishing at site. This difference is even larger when you consider electro-fishing and grouping species in species group comparable to eDNA, showing the better efficiency of sampling of species detection with eDNA in comparison with electro-fishing. Several species of high interest for conservation are only or mainly caught by eDNA method. For example, the sterlet, the sturgeon Accipenser rutenus, a very endangered species, was never caught by traditional electro-fishing. At the opposite, the eDNA survey shows its presence regularly up to Vienna, 1,900 kilometers upstream from the Black Sea. It has been also detected in one tributary, the Tisa River, where you have the black triangle. A second example concerns the two shad, Alosa immatriculata and Alosa pontica, which are two anadromous species entering the Danube from the Black Sea for reproduction each year in spring. Our DNA study shows that they are present all along the lower Danube, around 1,000 kilometers, but that their migrations are blocked at the dams of the Iron Gate, which is the first big obstacle for long migratory fish species on the Danube. These species are detected 5 times less by electro-fishing than eDNA. A last example, the single straber, a benthic fish, is detected by eDNA regularly on all along the Danube river course, from downstream to upstream, and also in half of the tributaries. At the opposite, it appears as a rare species in the electro-fishing catch. That's a general trend. The benthic species are much better detected by eDNA because of the limitation of electro-fishing method in large river, where only the shoreline is sampled. The number of reads per species gives also a clear picture of the relative abundance of species. You can follow here the longitudinal distribution of the different species from the source of the Danube to the Black Sea. Upstream, the typical species are, for example, the minnow, the stone loach, the grey line, followed by the chub and the cactus chub, an endemic species of the Danube, Rutulus virgo. Some species are present all along the river, like the bleak, the roach, the barbel, or the bream, for example. The last species group are more abundant in the downstream part, like the rude, Carassius, the shads, or the spined loach. The stellate sturgeon was only caught in the Danube delta, and this general distribution is quite in agreement with the previous results obtained with traditional electro-fishing methods. This eDNA data can also be used to assess the ecological status of the Danube River. Here we computed a fish assessment method using metrics describing functional traits of the species in relation with their habitat or their sensitivity to oxygen depletion in water. This method has been used to intercalibrate the river fish assessment methods employed for the Water Framework Directive at the European level and also in the Danubian intercalibration group. Using eDNA, most of the sites are classified as moderate, with several sites classified as poor in the lower Danube at the vicinity of the Iron Gate, and the better status are close to the Danube delta. When comparing this data with electro-fishing data using the same assessment method, 6 sites have the same ecological class, or 7 sites are only different for one ecological class. In conclusion, eDNA appears as a promising tool for the ecological assessment and the survey of rare or endangered species from the Danube, and to assess regularly the ecological status of the river. Thanks for your attention.